In this video, we'll be looking at this kangaroo taking care of a baby hippo. Also, we'll be doing some more multiple choice questions on the topic of eukaryotic and prokaryotic cells. So let's just get into it. Always try and give it a go by yourself to see if you already know what's going on. So what is a role of cholesterol in animal cells? So again, we know that there are prokaryotic cells and eukaryotic cells. Eukaryotic cells apply to animal and plant cells, so us human beings included. And then prokaryotic cells apply to small microbes like bacteria and things like that. So we also know that eukaryotic cells can be divided into animal and plant cells, right? Animal versus plant cells. Now, one of the differences is cholesterol. Animal cells have cholesterol, so there are many differences like this. We can see many differences that I made in a previous video. One of them being this. Cholesterol. Animal cells have cholesterol, whereas plant cells do not have cholesterol in their plasma membrane. So what is the purpose? So here we have an animal cell and this membrane, this plasma membrane. We know its structure looks like this, the membrane. It's made up of two layers, a phospholipid bilayer. These phospholipids, two layers of them upside down, facing each other. And in animal cells, there is also another thing called cholesterol that is arranged in between these little phospholipids. And basically, what it does is it enhances the fluidity. It enhances the bendability of the membrane. So if there's a lot of cholesterol, the membrane is able to bend more, to be more fluid, to be more wiggly, you know what I mean? So it enhances the bendiness of a membrane, basically. It makes it more bendy. So plant cells do not have this. So therefore, plant cells are more rigid, more hard to bend. They're more firm compared to animal cells that can bend and shape and have different shapes. So that's the purpose of cholesterol in a plasma membrane. So automatically, just by knowing that what cholesterol does, you can just directly get to the answer. It controls membrane fluidity. All of the other options are going to be random statements that may not even apply to what cholesterol does, just to try and throw you off. So if you know what cholesterol does, this question is pretty straightforward. The rest of the answers will just try and throw you off or confuse you. So next one. Which structure found in eukaryotes has a single membrane? Okay, this question I remember when I saw this for the first time when I was doing IB recently. This I have no idea how you can know this. You just need to memorize this, I guess. So try it by yourself first. So basically the answer for this one is lysosome. So nucleus, chloroplast, and mitochondria have double membrane, okay? Two membranes. So for example, let's look at here. Basically this thing right here has two membranes. What I mean then is by two membranes is it will have one and then another one under it. So it has two layers of membrane, whereas a plasma membrane, you can see, only has one layer. Okay, so the nucleus, the mitochondria, and the chloroplast will all have a double membrane. Okay, this you just need to memorize. Okay, because there are various other ones that are not double membrane, so single membrane, including lysosome, vacuole, endoplasmic reticulum, Golgi apparatus. So basically all the other organelles. So if we look at this image here, this one here, this one here, this one here, and the vacuoles, so this one, will all have single membrane, okay? So this is something, again, that I think is pretty hard if you don't, if your teachers haven't taught you about this. So just, I would recommend memorize this, because there's no real way of knowing this out of nowhere, right? Okay, here's the next one. The following electron micrograph shows part of a palisade mesophyll cell. Which of the labeled structure controls the exchange of substances to and from the cell? Okay, so here we have, we know an electron micrograph is what is used to be able to see organelles. So what we're looking at here is one organelle in a cell. So this is not a cell itself. This is an entire organelle, and we know, if you know from experience, by looking at enough pictures of this organelle, you'll know this is a chloroplast. Okay, so this is in a plant cell. And already, just by knowing that a palisade mesophyll cell is a plant cell, you'll know that this is a plant cell. Because this is the only cell that you need to know that is a plant cell. This is in the book, palisade mesophyll cell. So as soon as you see this name, you know they're talking about plant cells. Okay, and basically a good way to remember that this is chloroplast is that it has all these lines going from one point, you can see, kind of directing to the other point. It like seems like there's one line here, another line, a bunch of lines going across from this one edge to this other edge. Okay, that's how it's going across. That's kind of the feeling you should get. It's like one continuous line here, here. That's what a chloroplast looks like. That's how you should recognize it, because a mitochondria will have a wiggly line like this. You see, that's a mitochondria. A chloroplast is definitely like this. There's no wiggly lines up and down, so it's not a mitochondria. So they're asking which of the labeled structures, A, B, C, or D, controls the is a structure that controls the exchange of substances to and from the cell. So we know the structure that controls the movement of substances into and out of the cell is the plasma membrane, right? We know it's the plasma membrane. So here, A would be the membrane of the chloroplast. So it's not this, okay? This only controls the movement of substances into and out of the chloroplast, not into and out of the cell. Okay, structure B, this is the cell wall. So this one does not control the movement of substances into and out of the cell. Okay, so what I'm talking about is this. So here we have the chloroplast. There's its membrane, and then the next membrane would be this, and this is the plasma membrane. And then the next membrane is the cell wall. So the cell wall is the most outer layer. And that's why I was saying, you can see B here is the most outer layer, and therefore it is the cell wall. And all that it does is control structure and stability. It doesn't have anything to do with the movement of substances into and out of the cell. We know the plasma membrane does that. And we know the plasma membrane is the layer right under the cell wall. So if this is the cell wall, this is the plasma membrane. So if we know this is the cell wall, then this layer C here must be the plasma membrane, and this will be the structure that controls the exchange of things into and out of the cell. Okay, because we can see here D is something inside. They're pointing to a structure inside of the chloroplast, and any structure inside of the chloroplast will have nothing to do with movement of substances into and out of the cell. The structure will be involved in generating, it will probably be involved in photosynthesis, right? Because it's a chloroplast, which helps make energy from light in a plant. Okay, this was also a very tricky question. I would just recommend looking at a lot of electron microscope images to get used to how organelles look like under electron microscope. Otherwise, you look at this and you don't even know what organelle this is, and you get very confused. It helps a lot to know how the organelles look like, because that gives you a big clue as to what's going on. Because normally, the chloroplasts are located right next to the membrane, and that's what gave me the idea that this is probably the membrane. Because look, see, they're almost always located right next to the membrane. This is obviously a cartoon, so normally the chloroplasts are not located there. They're almost always located right next to the membrane like this. Okay, next one. Which organism has DNA located in 3 organelles? This one, again, very tricky question because it involves multiple topics, but with your knowledge on prokaryotes and eukaryotes, you should be able to get this regardless. Okay, so you're going to go through this and see. A sponge, what the heck are they talking about? Because this is related to another topic. A fern, okay, you should know a fern is a plant cell, is a plant, meaning it's made of plant cells. So do plant cells contain any organelles that have DNA located in them? So let's go take a look. Here we have a plant cell. We know one organelle is the nucleus that has DNA in it, right? So that's one organelle. We also know mitochondria has DNA in it, right? So that's 2. And the last one would be a chloroplast. So there's 3 organelles that have DNA inside them, because an animal cell only has 2, a mitochondria and a nucleus. So a plant cell is very unique as that it has 3 organelles with DNA inside it. The chloroplast, the mitochondria, and the nucleus. All 3 of these. All 3 of the organelles that have a double membrane layer, as we just did the question before. All 3 of these have double membrane layers. double layer membranes, I mean. So we know it's B because you'll go to C and be like a flatworm. Okay, I haven't learned about this yet. Yeah, because this is in another topic. Maybe you'll go to D then and you'll know a bacteria. Oh, I know bacteria is prokaryotic cells, and I know prokaryotic cells don't actually have organelles. Therefore, okay, it can definitely not be D. Okay, so this is a tricky question, but if you know what a fern is a plant, then you should be able to get this question. But it is a hard one because it's tricking you with these topics that you haven't learned yet. So here we go, last one. Which organelle is involved in generating vesicles destined for the cell membrane? Okay, let's go take a look. So here we have, we'll use the plant cell because it's clearer. So here we have the nucleus, and we know DNA is a code to make proteins. So the code will go outside the nucleus to the rough endoplasmic reticulum. This rough endoplasmic reticulum is where there are some ribosomes, these small little dots. These dots will take your DNA, read it, and make a protein according to what the DNA is asking. The ribosomes will read your code, it will read your DNA and make a protein according to that code. After it makes the protein will move to the smooth ER. The smooth, so it'll move from the rough endoplasmic reticulum down to the smooth endoplasmic reticulum, and then the smooth endoplasmic reticulum will send it to the Golgi apparatus through a vesicle. So basically, the protein will go into a vesicle, travel all the way down to the Golgi apparatus. So technically here, a vesicle was made, but this vesicle was not sent to the cell membrane. It was sent to another organelle, so it can't be the smooth endoplasmic reticulum, right? So once the protein arrives in the Golgi apparatus, it will modify it, package it, and all those kind of things, make some small modifications to your proteins, and then it will send those proteins in a vesicle to your cell membrane so that it can be sent outside the cells to other cells or just simply inside your membrane, okay? So it has to be Golgi apparatus because the rough endoplasmic and smooth endoplasmic reticulum, the rough endoplasmic reticulum makes the protein, it sends it to the smooth endoplasmic reticulum, and the smooth endoplasmic reticulum will send it through a vesicle to the Golgi apparatus, not to the cell membrane. That's why it cannot be the smooth endoplasmic reticulum. And the lysosome does not make vesicles. Okay, so this organelle is just not related in making vesicles. So I hope these questions helped you somewhat. They are a bit difficult, so I would just recommend getting more and more practice and making sure that you covered all the knowledge related to this chapter before you continue to do more questions.